Our immune system is one of nature's most powerful weapons, built to destroy anything that threatens us. But what happens when that turns inward? For decades, scientists have believed that a gland called thymus played the major role in preventing this. But what if there were undiscovered guardians patrolling every organ and preventing internal sabotage? This is the story of three scientists, Sakaguchi, Brunkow and Ramsdell, who have uncovered those very breaks, the regulatory T cells, and why their work has just been awarded the 2025 Nobel Prize in Medicine. Join me as we trace their journey, unravel the secrets of immune tolerance, and peer into the future of medicine. Every second, our immune cells patrol billions of our own cells and decides who is friend or foe. In medical school, we are taught that the thymus, the organ where T cells mature, acts like a screening center. The T cells that react against our own cells are mostly eliminated before they enter the body circulation. That negative selection explains a lot, but not everything. If this process was perfect, autoimmune diseases would have been vanishingly rare, but it isn't. Clinicians saw patients in whom the immune system attacked the pancreas, the gut, the skin, or the joints, and basic experiments showed that some self-reactive cells nevertheless escaped thymic deletion. That gap in the story suggested that there might be some other layer of control, a way the body keeps potentially dangerous T cells in check after they leave the thymus. Working in Japan in the late 1980s and early 1990s, a scientist named Shimon Sakaguchi began to suspect such a hidden force existed. In one of his colleagues' work, they removed the gland in mice to establish the function of thymus in immunity. But instead of reducing the T cell number and reducing immunity, they found a contradiction. There was a range of autoimmune effects taking place in these mice. Sakaguchi asked a simple question. If removing the thymus sometimes caused runaway autoimmunity, could some cells in the periphery actually prevent that runaway? He found those cells when he transferred normal T cells into those mice. Something extraordinary had happened, and the autoimmunity vanished. These mysterious cells, expressing a molecule of CD25, acted like brakes instead of killers. Sakaguchi's experiments gave us the first clear evidence that a dedicated population of suppressive T cells exists, which he called regulatory T cells. At first, many dismissed the idea. It challenged the dogma that deletion alone maintained tolerance. But across the Atlantic, a different thread of story was coming into focus. For decades, geneticists had observed a strain of male mice known as Krufi mice, with a devastating early onset inflammatory disease. These animals developed severe multi-organ inflammation and died young. It was also inherited. Brunkow and Ramsdell dug deep into the genome to find the culprit. Although it is now possible to map the entire genome and search for a mutated gene in a mice in a span of few days, in 1990s, it was like looking for a needle inside a gigantic haystack. For giving you a perspective, the X chromosome in a mice has around 170 million base pair nucleotides. After years of detailed mapping and searching, in 2001, Mary Brunkow and Fred Ramsdell mapped that defect to a gene they call FOXP3, a forkhead box transcription factor. They showed that mutations in this gene produce the scurvy phenotype in mice. They even suspected a human equivalent of the gene and eventually matched mutations found in children with IPEX syndrome, a severe human immune dysregulation disorder found in infants. A couple of years later, Sakaguchi and other scientists were able to prove that FOX3 gene controls the production of regulatory T cells, providing the missing molecular link that proves the presence of a peripheral immune tolerance. FOXP3 gene produces transcription factors that defines the regulatory T cell lineage and enables their suppressive behavior. When FOXP3 is absent or mutated, that program fails and immune restraint collapses. This was a conceptual pivot. Immune tolerance was not just solely produced by removing dangerous cells in the thymus, but it also depends on active suppression in the periphery through cells that are controlled by FOXP3. The arc of discovery here is worth pausing on. Sakaguchi's experiments pointed to a population of cells that could silence immune attacks. Brunkow and Ramsdell's genetic detective work revealed the FOXP3 switch that gives those cells their identity. Together, they shaped our understanding from that of a central immune tolerance to that also requiring a peripheral immune tolerance. That conceptual shift has opened a new field of research and seeded a generation of therapeutic strategies. For this integrated discovery of the cellular and molecular foundation of immune restraint, they received the Nobel Prize in Medicine in 2025. So what exactly do these cells do? Some regulatory T cells are born in the thymus, which are called natural regulatory T cells. Others are formed later in peripheral tissues when the immune system encounters antigens in the presence of signals like TGF-beta and interleukin-2. Once active, they calm the storm. They secrete anti-inflammatory cytokines, interleukin-10, TGF-beta, interleukin-35 that tell other T cells to stand down. They consume interleukin-2, depriving effector cells of fuel. Through molecules like CTLA4, they silence antigen-presenting cells and prevent further activation. And by generating adenosine and modifying dendritic cells, they maintain a peaceful immune microenvironment. Without regulatory T cells, autoimmunity flourishes. With too many or too powerful regulatory T cells, cancers may hide behind their protection. Today, scientists are testing new ways to transfer the regulatory T cells for treatment of autoimmune disorders like type 1 diabetes, multiple sclerosis, or lupus. In transplantation, the therapy may one day replace harsh immunosuppressants, teaching the body to accept a new organ as its own. In cancer, the goal flips to disable or reprogram the regulatory T cells that shield tumors from immune attack. Engineers are even designing a chimeric antigen receptor, which are genetically tailored suppressor T cells that can hone precisely to inflamed tissue. As we look forward, the questions feel both practical and philosophical. How precise can we make regulatory T cell based therapies so we suppress autoimmunity without leaving patients vulnerable to infection or cancer? Can we reliably stabilize engineered regulatory T cells so they do not lose FOXP3 and convert into inflammatory cells? And at a deeper level, what other balancing system written in our genes or in the cells quietly maintain our internal homeostasis? The work honored by the prize does not close a book so much as it opens a library of new possibilities. Thank you for watching, and if you enjoyed the video, don't forget to subscribe and drop a like.